Hey, I'm Wadi, and this is 10 questions about your video. Your video? Your video. Yo. So after releasing a video about a complicated scientific topic, usually we get a lot of questions about points that were not clear, about some technical aspects that we did not get the chance to explain, without the video turning into a 30 minutes lecture. Therefore, we decided to create this approach, more like a Q&A session, in order to address these questions and try to answer them. Because learning is not a one-way street. It's more like a give and take. Starting with question number 1. You tested bone implants and showed us which one might be the best. Which materials did you use in those implants and why? Well, we used a couple of ceramic materials. And these materials are called beta-tricalcium phosphate and hydroxyapatite. And they look like this. White powder, non-sniffable. They look kind of similar. The differences between these two, hydroxyapatite and beta-tricalcium phosphate, is in the chemical composition. And when we implant these bone grafts in the body, these ceramics resorb with time, so they disappear. And the body creates its own native tissue that replaces those implants so that the body can regenerate by itself. So the implants would disappear or resorb with time, and the body would create its own bone as a replacement. Question number 2. How long does it take before we could use such implants in our bodies? It takes a while. The thing is, first we start by testing in vitro or in the lab. This is similar to what we did in the previous video. Now, after we do this, we identify optimal implants or find what is the best for us to research on further. And then we do animal trials, hopefully ethically, although there's a lot of debate. Although there's a lot of debate about the ethical side of testing on animals, but we still need to do that, unfortunately. And after animal trials, we run multiple stages of human trials, and all this is governed by the European Commission. So if you're interested in more information, you can read the document called the Commission Directive 2003/94/EC. It has a lot of information about the procedure. Number 3. In the test protocol, you crush the implants and kill the cells. Is this ethical? And were any animals tortured in the course of this experiment? Ethics. No animals were tortured in the course of this experiment. We had to kill the cells to extract the RNA. You've probably seen the DNA, which is a double-stranded structure. The RNA is single-stranded structure, and it holds some of the genetic information of the cell. So we needed to kill those cells in order to extract the RNA, which is stuck inside of the center part of the cell. But if you think about it, in the food industry, in the dairy, and the meat industry, they're killing millions and millions and millions of cells every day for us to have a nice piece of steak for dinner. So the number of cells that we had to sacrifice for this measurement is very marginal compared to what the food industry is killing almost every day. Just keep this in mind. Question number 4. You showed us a fridge to store the samples at -80 degrees C. Do cells survive such a low temperature? No, they don't. Ice crystals rip the cells apart when they expand, when they grow. And so no cell really survives the freezing process. And the thing is, thawing is the problematic stage. It's not the freezing process, it's actually bringing it down to room temperature. This is why cryopreservation is still not a feasible solution, for us to store our bodies in liquid nitrogen for decades and then revive our cells in the future, because this thawing stage is still destroying the cells, which is, killing us. Question number 5. I thought that cells are round in shape. Why the cells you showed have longitudinal alignment? Yes, cells change shape when we put them in a medium or when we culture them inside of a flask. And this depends on the microenvironment surrounding those cells. And they change in an adaptation mechanism and in order to communicate with each other better. Nature is fascinating, isn't it? Question number 6. You use a dye called LMR blue. What does it do exactly? Well, LMR blue is a dye that is oxidized in its original form into a blue color. And when it's reduced, so when it undergoes a reduction reaction, it turns from blue to pink or red in color. And actively metabolizing or viable cells, or basically living cells, induce these reduction reactions that turn the dye from blue to red or pink in color. And so the more alive and active and breathing that cell is, the larger the change in color that we observe in this dye. And so we can measure this degree of change in color and using a spectrometer, of course, and we can correlate that with the level of viability or how much the cell is breathing. Question number 7. Who was filming you while you were doing the measurements? Well, my assistant was filming me the whole time. And she's my colleague, and she's the expert in biology. I'm not. So she was teaching me, she was helping me out, and at the same time filming. She's fantastic. Number 8. What is the aggressive solution you used, and what does it do? Yes, you mean the Piranha solution, right? The Piranha solution is a very corrosive solution that attacks the organic materials on the surface of the implant. And this causes etching to happen on this outer surface of the implant, which changes the micro roughness and the texture of the implant. And we noticed from the measurements last week that the cells react differently towards such implants with a specific surface roughness. So the surface texture and micro roughness does change how the cells react. Question number 9. You were wearing different color gloves. Is there a difference between them? Nope, there is no difference between them. I just found the right size and put it on. It happened that they had different glove boxes. Question number 10. Why were you wearing a medical mask in one of the scenes? Why do you have to ask? No, actually because I was using a chemical called diazole or pyrozole, some people call it. So it's a chemical that stinks and something you don't want, to end up in your lungs. So I just found a mask and I put it on. So thank you so much for your questions. If you have more, I'll be around. Just ask. And I'll try to keep you informed about what happens in this field and the latest advancements in research when it comes to bones. Have a good time.